So now, I'm going to show you how to check the air temperature. First, hold the thermometer at eye level and check that the mercury line is below the scale. Secondly, leave it for 2 to 3 minutes to stabilize. Then, read the temperature at eye level where the mercury line stops. And now you can record the reading.
I'm going to show how to measure soil temperature using a mercury thermometer. First, choose a spot on the soil that represents your study area. Gently clear away leaves, stones, or mulch so the soil surface is exposed. This helps you get a true temperature reading. Next, take your mercury thermometer and place the metal bulb or sensing tip directly on the soil surface. Make sure it has full contact with the soil. If you want the surface temperature, avoid pushing it deep and just let the tip sit on the top layer of soil. Now, wait until the reading stops moving. Once the needle or column becomes steady, read the temperature at the eye level on the scale. Record it along with the time. Gently lift the thermometer from the soil. Lastly, wipe it clean so it is ready for the next use.
How to use a soil moisture meter. Make sure the prop is clean and dry, then insert the metal prop into the soil until it reaches the root zone. Don't push it too hard to avoid bending the prop. Hold the meter and wait for 5 to 10 seconds. Then, the digital screen will show the soil moisture percentage.
Now I'm going to explain how to measure light intensity using a portable light meter. Firstly, we start by switching on the portable light meter. Then, hold the sensor directly towards the light source, but wait for a few seconds until the reading becomes steady and record the results.
So this is how to use the chlorophyll device. First, place the leaf flat between the clamps and gently press them together. Now, you have to wait for a moment and the meter will display the chlorophyll value on the screen. So this is comparison of environmental conditions and plant species in both ecosystems. The environmental condition of Arboretum is shadier, more humid due to presence of tall trees. Meanwhile, Takasago has open space, higher sunlight exposure, and the soil is still pretty much developing organic layer. The most dominant plants at the Arboretum is native forest tree species that are slow-growing hardwoods, while the Takasago is dominated mostly by pioneer shrubs and fast-growing plants which is suitable for early reforestation.
Why are plant species in both ecosystems different? The plant species are different because each ecosystem is at a different stage of development and has different objectives. Arboretum focus on conservation of native and high-value tropical forest species. The trees there grow slower and require long-term maintenance, which contribute to biodiversity preservation, while Takasago is designed as a reforestation and environmental educational site. So it uses pioneer and fast-growing plants that can tolerate high sunlight and exposed soil. grow quickly to stabilize the area, also improve soil condition for future forest trees.
The conclusion is the Arboretum is a mature ecosystem with slow-growing native tree, while Takasago is still developing and uses fast-growing pioneer species. These choices match their purposes. The Arboretum focuses on long-term conservation, and Takasago focuses on quick greening and education. Together, they support Unimar's goal of sustainable forest restoration.